Glycolysis is one of the main metabolic pathways. It breaks down glucose, so let's break it down. We'll start with just a quick overview, and then we'll go into the chemical logic as the various steps. So glycolysis, the breaking down of glucose to give you pyruvate. This pyruvate can then, in the presence of oxygen, be taken through oxidative decarboxylation to give you acetyl-CoA, which can then go through the citric acid cycle, get oxidized further. All the electrons that you're generating from those oxidation reactions that you're passing off to electron carriers, those electron carriers can go and take those electrons through the electron transport chain to give you ATP. In the absence of oxygen or the absence of the electron transport chain for some reason, what's going to happen is that you are going to need to regenerate NAD+ to keep glycolysis going, and you're going to need to keep NADH from building up. So as an alternative to that aerobic respiration, that aerobic glycolysis, what you can do is you can convert the pyruvate to lactate, which is going to be using NADH, regenerating NAD+, and that's going to allow you to keep glycolysis going even in the absence of oxygen. So that's anaerobic respiration. Both of them are starting to start with this central part of glycolysis, this glucose to pyruvate, and so that's where we're going to start. Note that the reverse pathway isn't truly a reverse. So we have glycolysis is these green arrows, and then gluconeogenesis is these blue arrows. So blue arrows are gluconeogenesis going in this direction, and this is glycolysis going in this direction. Gluconeogenesis is the making of glucose from non-sugar sources, and so it's in its entirety, it goes pyruvate to glucose. It uses 7 of the same enzymes as glycolysis. You can use the same enzymes for these easily reversible reactions where we can control the direction just by controlling the concentrations on either side. But for these steps that are metabolically irreversible, these one-way arrows, we have to take detours around them in the process of gluconeogenesis in order to ensure that the pathway is going to be favorable energetically in both directions. We're going to actually have to spend an ATP and a GTP in order to do gluconeogenesis. We're going to have an extra intermediate, this oxaloacetate, and we're going to have one more enzyme because we have one more intermediate. And so we'll talk about the glycolysis, the gluconeogenesis, the steps that we have to detour, and the chemical logic of each of these steps. Let's go. Glycolysis can be broken into 2 main phases, the investment phase and the payoff phase. In the investment phase, you're actually going to be spending 2 ATP. This is going to be in order to trap the glucose inside the cell, commit it to glycolysis, and prepare for the payoff phase. The payoff phase is going to happen twice because you're going to split the 6-carbon compound into 2 3-carbon compounds and then do everything down here 2 times. As a net result, you're going to generate 2 ATP and 2 NADH, and that NADH, assuming you can use the electron transport chain in oxidative phosphorylation, is going to actually give you the bulk of the energy that you can get from glycolysis, the bulk of the ATP that you can generate. And then most of the ATP that you're generating from the glucose, however, is actually going to be from NADH from the citric acid cycle. But for now, we're focusing on glycolysis. So let's start from the top. The top step of glycolysis is not specific for glycolysis. Instead, the purpose of the first step, the hexokinase step, is really just to trap the glucose inside of the cell. No matter what you want to do with that glucose, you got to keep it from leaving the cell. And so you take in the glucose through a glucose transporter, a GLUT transporter, typically, and then what's going to happen is that you've got this glucose inside the cell, but to keep the glucose from leaving the cell, you need to trap it. And the way that you're going to trap it is with hexokinase phosphorylating it. Hexokinase is going to be a traditional kinase in the sense that it's going to transfer a phosphoryl group from ATP onto your substrate, so in this case glucose, and specifically in this case the 6th position of the glucose. I say phosphoryl group because what you're transferring is this phosphorus with 3 oxygens. The 4th oxygen of this phosphate group is actually coming from the glucose itself. And I mention this because this is not an oxidation reaction because this carbon is still bound to 1 oxygen here, and it's still bound to 1 oxygen here. It's not increasing its oxidation number, whereas we will see, and there's a step down the line, this glyceraldehyde 3-phosphate dehydrogenase step, where we actually are going to be changing the oxidation number, and then we are going to be needing NAD+. But here we're just transferring the phosphoryl group, and so we don't. Note too that this is going to leave us with ADP, so we're not going to have a plus PI here because the phosphate group, rather than just being released free floating, is actually getting transferred onto the glucose. So we take the phosphoryl group off of ATP, stick it onto glucose, that gives us glucose 6-phosphate, and we've trapped the glucose inside the cell. We've really trapped it in tissues that aren't our liver and our kidneys, because pretty much only your liver and your kidneys, small amounts in other tissues, have this enzyme glucose 6-phosphatase, which catalyzes the kind of opposite reaction in gluconeogenesis. So these are one-way reactions in gluconeogenesis and glycolysis. In glycolysis, you transfer the phosphate group, or technically the phosphoryl group, from ATP onto the glucose to give you glucose 6-phosphate. In gluconeogenesis, rather than trying to just simply reverse this, what you do is you actually just use water to hydrolyze off that phosphate group. And this is going to be an energetically favorable reaction, whereas trying to put the phosphoryl group back on ADP directly would not be favorable. And so you use these different enzymes, and so you have glucose 6-phosphatase in your liver and your kidneys that are then able to free the glucose to be released. In your other tissues, even if they could do gluconeogenesis up until this point of glucose 6-phosphate, they can't actually remove that phosphate group, and so they can't release the glucose into the bloodstream. So your liver and your kidneys are going to be the two main tissues that are responsible for releasing that glucose. You might be wondering, well, what about your pancreas? Your pancreas is really important for sensing the glucose and controlling the hormones that help control whether the glucose is released from your liver and your kidneys, the insulin and the glucagon. Your pancreas, therefore, needs to be able to sense the levels of glucose, and your liver also needs to be able to sense the levels of glucose and to be able to take in excess glucose from the bloodstream and store it as glycogen and as fat. And so what's going to happen is that in your liver and your pancreas, you have this version of hexokinase called hexokinase 4, the isoform called hexokinase 4, which we typically refer to as glucokinase. And glucokinase is actually not going to be inhibited by glucose 6-phosphate, so it can keep trapping the glucose inside the cell, even if the glucose levels are high. Whereas in other tissues, to prevent them from taking in too much of the glucose because they can't let it out if they trap it, then they're going to have their hexokinase inhibited by the glucose 6-phosphate that they bring in. Your liver and your pancreas are also going to have a lower affinity hexokinase so that it doesn't hog everything. So this is an important step for getting the glucose trapped inside the cell, but it's not committing it to glycolysis, just committing the cell to keeping it. There are still other options, and some of them you can go to directly from glucose 6-phosphate. You can go directly through phosphoglucose mutase in order to get into the glycogen pathway, through glucose 6-phosphate dehydrogenase to go to the pentose phosphate pathway, or you can go this isomerization in order to get fructose 6-phosphate, which has additional fates such as the hexosamine pathway and making F26BP. So this is an isomerization reaction. With an isomerization reaction, you're just shifting the position of functional groups or shifting the position of atoms within a molecule. If we look at the structures of glucose 6-phosphate and fructose 6-phosphate, at first we might be like, oh, redox reaction. We're going to be going from a carbonyl to a hydroxyl group. That'd be a reduction. We're going to need NADH. Well, not so fast, because if we look at the second carbon position, well, now we're here going from a hydroxyl group to a carbonyl group, so that would be an oxidation. So we have this kind of internal redox reaction, but overall, we're not going to be changing our oxidation state, and we're not going to be needing any reducing equivalents in the form of electron carriers like NADH or NAD+. And what's the purpose of this, you might ask? Well, if we think about this, we freed up a hydroxyl group. And as we saw before, a hydroxyl group can actually grab a phosphoryl group from an ATP in order to get phosphorylated. And so that's what we're going to see in the next step. So in this step, the phosphofructokinase 1 is actually going to take a phosphoryl group and transfer it onto the fructose 6-phosphate at that one position that we just freed up to give us fructose 1,6-bisphosphate. And this is what we consider the committed step of glycolysis. Before, we had that isomerization reaction. Isomerization reactions are typically readily reversible, so we could go easily back and forth and we hadn't committed ourselves to any one fate with that glucose that we trapped. However, once we go and we phosphorylate it, now we kind of committed it more to this glycolysis path, so we consider this the committed step of glycolysis. As such, we're going to regulate it very highly. And we're going to need to use a different enzyme in the reverse pathway because this is going to be one of those very energetically favorable reactions that we can't just easily go back from. It's going to be one of those metabolically irreversible reactions. So in gluconeogenesis, we have fructose 1,6-bisphosphatase 1, and what it's going to do is it's actually just going to chop off the phosphate group with water, similarly to what we saw with glucose 6-phosphatase. For now, let's keep on going in our glycolysis direction. So we've got this F16BP, this fructose 1,6-bisphosphate. And again, before we had more fates, but now we're kind of thinking about glycolysis. If you're wondering, why do we do this? Why do we phosphorylate the second end? Well, let's think about it. Now what's going to happen is that even if we were to break this molecule in two, each part would have a phosphate group. And thus each part would kind of be stuck inside the cell and kept from floating out because you've got that charge group on it. Additionally, if we split this directly in half, we're going to end up with two pieces that are going to be interconvertible between one another. So now that we've got it phosphorylated on both ends, we can go ahead and do that splitting. We're going to take the 6-carbon compound and we're going to split it in half in a retroaldol reaction. And so we call this an aldolase. Its full name is fructose bisphosphate aldolase, but typically we just refer to it as aldolase. And it's going to break that 6-carbon into two 3-carbon parts, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It's really helpful in order to convert these bigger pieces into smaller pieces because these smaller pieces can be used for more pathways, more things, and you can just do the same process twice with the smaller pieces with the same enzymes. But then you can ship the smaller pieces off in different directions. For example, the dihydroxyacetone phosphate can be used to make glycerol. However, the dihydroxyacetone phosphate cannot continue on directly into glycolysis, but the glyceraldehyde 3-phosphate can. And the two of these are going to be directly convertible just by isomerization. So when you split it, you get these two pieces. So this is the dihydroxyacetone, and this is the glyceraldehyde 3-phosphate. Note that we've got the aldehyde group here. Here, it's just shown it flipped over so that you can see more easily that all we're doing is one of those isomerization reactions similarly to what we saw before. Because this reaction is readily reversible, but because only the G3P can actually keep going through glycolysis, if you take the G3P through glycolysis, it's going to draw this whole pathway forward and through these steps. But you can go in either direction. But if you're going down glycolysis, this is going to be shifting this reaction this way, and so you're going to be drawing all of the DHAP into this part. And now everything that's going to happen further down is going to be repeated twice, once per 3-carbon piece that we had. So once directly from that glyceraldehyde 3-phosphate, and then once from the glyceraldehyde 3-phosphate that we get when we convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And we do that conversion using this triose phosphate isomerase. And now we're in the energy payout stage. And the first energy payout we get is actually going to be indirect. We're generating NADH at this step, not ATP. But remember that NADH, as long as we have the OXPHOS going, oxidative phosphorylation, we can do our electron transport chain, then we're going to be able to generate ATP from that. So in this reaction, we have glyceraldehyde 3-phosphate dehydrogenase, and it's going to be taking glyceraldehyde 3-phosphate and making 1,3-bisphosphoglycerate. This is going to be adding a phosphate group, and it's going to be adding an entire phosphate group rather than just a phosphoryl group. And so if you look at this carbon here, it has 2 bonds to an oxygen and 1 bond to a hydrogen. If we look at 1,3-BPG, we have 3 bonds to an oxygen, and we've lost a bond to a hydrogen. So we've gained bonds to oxygen, lost bonds to hydrogen. This is an oxidation reaction. For oxidation reactions, we're typically going to be using NAD+. And so we're going to need NAD+ in order to oxidize this. And we're going to need a phosphate group in order to actually get the phosphate group to put on here. And so rather than coming from ATP, where if you were to remove a phosphate group, you would be left with the kind of weird partial molecule for the ADP, we're going to actually just take an inorganic phosphate, a free-floating phosphate, and stick it on. This is probably one of the most confusing steps of glycolysis because normally when we think about a reaction that's putting on a phosphate group, we think about a kinase that is using ATP, and it's going to be an energetically favorable reaction, a highly energetically favorable reaction that you can't just go back from easily, such as we saw with our hexokinase step and our phosphofructokinase step. However, this reaction is actually going to be reversible, and it's going to be an oxidation reaction. Instead of using ATP, we're using this inorganic phosphate, and we're using NAD+. And we're going to be oxidizing and adding a phosphate group. And you can see that we really are oxidizing it because if we remove that phosphate group, we're left with a carboxyl group. And so this carbon is still left in a more oxidized state than before. In this step, what we call this type of phosphorylation is oxidative phosphorylation. So typically when we think about oxidative phosphorylation, you're typically thinking about OXPHOS, you're thinking about mitochondrial oxidative phosphorylation, where you're using the electron transport chain to pump protons into the intermembrane space of the mitochondria and use that proton gradient in order to power the synthesis of ATP. That too takes an inorganic phosphate, but then it sticks on to ADP to give you ATP. Here we're sticking the phosphate group onto glyceraldehyde 3-phosphate in order to give us 1,3-bisphosphoglycerate. So although it is an oxidative phosphorylation, it's not the oxidative phosphorylation that we typically refer to when we talk about OXPHOS. So why do we do that? Well, now we just set ourselves up to transfer the phosphoryl group from 1,3-BPG onto ADP to give us ATP. So in the OXPHOS in our mitochondria, we're taking a free-floating phosphate and sticking it onto ADP. Here we're taking a phosphate group that is going to be present on an organic molecule and sticking it onto ADP. This is an example of what we call substrate level phosphorylation. So we're taking the phosphoryl group from one molecule and sticking it onto another molecule. Specifically, we're sticking it onto ADP, and this is going to give us ATP. Note that no free-floating phosphates, no inorganic phosphates are involved in here. So we put the phosphate group from a free-floating phosphate onto the glyceraldehyde 3-phosphate, and that is going to give us this energetically favorable reaction where we can then transfer the phosphoryl group from that onto ADP to give us ATP and leave us with 3-phosphoglycerate. So we used an oxidative reaction to get the energy to put the phosphate group onto glyceraldehyde 3-phosphate. And then we used another favorable reaction in order to stick that phosphate group onto ADP to give us ATP. But we were really just sticking the phosphoryl group, and so we're left with this more oxidized compound, this 3-phosphoglycerate. This is taking place with phosphoglycerate kinase, which is another one of those confusing reactions because, again, we have one of these reversible reactions that is involving the phosphoryl group transfer. So you just have to keep in mind that these reactions are going to be reversible, but most kinase reactions you see are not going to be. But things can be a little more reversible when you're dealing with this substrate level phosphorylation and oxidative phosphorylation than when you're dealing with those simple kind of more hydrolysis-y-like reactions. Now we're left with this 3-phosphoglycerate, and we're going to do another isomerization. This one is going to involve phosphoglycerate mutase. And so phosphoglycerate mutase is going to take this phosphate group and just shift it to the center position. This might seem kind of weird, but it's going to set us up for the next step. And so we're just shifting this phosphate group to the middle, and this is going to free up here. It's going to then set us up so that enolase can do a dehydration reaction. So if we look here, we see that we have this carbon and it's attached to 2 hydrogens and a hydroxyl group. And here it's attached to no hydroxyl group, but 2 hydrogens. And now it's also formed a double bond to this carbon, which means that this carbon has now lost a bond to a hydrogen. So at first glance, we might think, oh, okay, we're going from a single bond to a double bond, and we're losing a bond to an oxygen. This must be an oxidation reaction. But If we think about what's going on, we're losing this oxygen, but we're also losing this hydrogen, and we're losing this hydrogen. So we're losing two hydrogens and an oxygen. Yep, that's our good old water. We're losing a water molecule. This is a dehydration and not an oxidation. If you think about the oxidation status of each of these, so this one, although it's losing a bond to an oxygen, it's not gaining a bond to a hydrogen. Instead, it's gaining a bond to a carbon, and this carbon is losing a bond to a hydrogen. So overall, this is not a redox reaction, it's a dehydration reaction, which is a form of elimination reaction. Because it's not redox, we're not going to be using NAD plus or NADH. And so now you might be wondering, why do we do that? Why do we go and dehydrate it? Well, what we've done is we've trapped ourselves in this form that's highly unstable. This phosphoenol pyruvate, if we think about an enol, that's where we have this alkene, so we have a double bond between two carbons, and then one of those carbons is a part of an alcohol. That's going to be really unfavorable, and it can tautomerize to form a ketone form that's much more favorable. And so, like the name suggests, phosphoenol pyruvate is going to have this phosphoenol form. The phosphate group is trapping it in this enol form. That's going to be highly energetically unfavorable. And so if you can go back to the keto form, then you're going to release energy. But in order to go back to the keto form, you've got to release the phosphate group. And you have so much energy from this reaction that you're going to be able to transfer that phosphate group onto the ADP to give you ATP. So this is another one of those substrate level phosphorylations. We're transferring the phosphate group from our phosphoenol pyruvate onto our ADP to give us ATP, and that's going to leave us with pyruvate. This is carried out by this enzyme pyruvate kinase, and it's a very highly energetically favorable reaction. So energetically favorable that for gluconeogenesis, we need to actually use two enzymes in order to do the equivalent of reversing it. And we're going to actually have an extra intermediate, this oxaloacetate, in the reverse pathway, spending an ATP and a GTP just to go back from pyruvate to phosphoenol pyruvate. So in that gluconeogenesis direction, you're going from pyruvate to oxaloacetate to phosphoenol pyruvate. This involves two enzymes. Pyruvate carboxylase is going to take you from pyruvate to oxaloacetate, and PEP carboxykinase, PEPCK, is going to take you from oxaloacetate to phosphoenol pyruvate. Now, this pyruvate carboxylase step has to take place in the mitochondria, but PEP carboxykinase can take place in the mitochondria or in the cytoplasm. And so you might be wondering, when are you going to do what? Well, let's think through this. In our gluconeogenesis pathway, we're going to require NADH for this glyceraldehyde phosphate dehydrogenase step. In glycolysis, we required NAD+, but in gluconeogenesis, we require NADH. All of this part of gluconeogenesis is going to happen in the cytosol, and all of glycolysis is going to happen in the cytosol. But this step in gluconeogenesis has to happen in the mitochondria, and this step can happen in the mitochondria or in the cytoplasm. In order to do gluconeogenesis, in order to maintain our redox balance, we need to make sure that we have NADH in the cytoplasm that we can use for this pathway. There's a complication, however, because oxaloacetate can't actually leave the mitochondria. And so if you want to do this PEP carboxykinase step in the cytoplasm, you're going to actually have to ship out malate and then convert that malate into oxaloacetate to then do this PEP carboxykinase step. So oxaloacetate is a member of the tricarboxylic acid cycle, this Krebs cycle, the citric acid cycle, whatever you want to call it. And it's really important. And it's really important that the levels of it are kept really low in the mitochondria, and that's not just able to go in and out of the mitochondria. So if you want to remove oxaloacetate from the mitochondria, you actually have to remove malate, which means that you have to reduce the oxaloacetate, which is going to generate NAD+. You take out that malate, then you undo the reduction. So you are going to oxidize the malate to give you oxaloacetate, and that generates NADH. And so if you do that, you're generating NADH in the cytoplasm where you have it ready to use. However, if you start with lactate, which we'll talk about in a minute, if you start with lactate, now you're going to actually produce NAD+ in order to generate pyruvate, which you can then take into the mitochondria and do all these steps with. And in that case, because you already generated the NADH out in the cytoplasm, you don't need to generate another one in the cytoplasm. And so you can do both steps, the pyruvate carboxylase and the PEP carboxykinase in the mitochondria, and then you don't have to worry about your redox balance. So why might you have lactate? You might have lactate if you need the NAD+. So remember here, we're talking about generating NADH for gluconeogenesis. But if we think about glycolysis, well, here we're going to need NAD+. We're going to need NAD+ in order to do this oxidative phosphorylation of the glyceraldehyde 3-phosphate. Now, if we are doing aerobic glycolysis, so we take our pyruvate and now we go and we use pyruvate dehydrogenase, we oxidize it further, give us acetyl-CoA, take that through the citric acid cycle. We take the electrons from those NADHs and we go and we put them through the electron transport chain and generate ATP. Great, that's going to regenerate our NAD+. And then that NAD+ can go and be used in glycolysis. And it's going to keep us from building up NADH. And that NADH was actually going to be able to inhibit enzymes of glycolysis. So it would inhibit our pyruvate kinase step. And it would inhibit enzymes of the citric acid cycle and things. So we want to keep the NADH from building up. And we want to keep the NAD+ high so that we can use it for glycolysis. If we can't do the electron transport chain, since we don't have oxygen, which would back up the whole chain because oxygen is going to be the final carrier in this electron transport chain. If we don't have the electron transport chain because we don't have mitochondria, such as our red blood cells, for any reason that we don't have the electron transport chain running, we need an alternative route in order to regenerate the NAD+. And so one way that we can do this is to anaerobically ferment. So in glycolysis, so we passed electrons onto NAD+ to give us NADH. Now we're going to pass those electrons back from NADH onto pyruvate, and that's going to give us the NAD+ that we need. The pyruvate is now lactate, and that lactate, we need to get rid of it. And so you can then ship it to your liver, and your liver can reverse the process. And when your liver reverses the process, they're generating NADH, and they can use that NADH in order to do gluconeogenesis to make glucose. And your liver, remember, it has this glucose 6-phosphatase, so it can actually release that glucose. So we have this process called the Cori cycle, where your muscles, if they're doing anaerobic respiration, and so they don't have enough oxygen, so or they can't use it, they can't do the electron transport chain fast enough, then what's going to happen is they're going to make lactate in order to keep NAD+ ratio high. They ship out the lactate that they make, take it to the liver in this Cori cycle. The liver converts it back to pyruvate, uses that pyruvate to make glucose, ships out the glucose to the muscles, and lucky muscles. One more time from the top. So glycolysis, we have the breakdown of glucose to give us pyruvate. It starts by trapping the glucose that enters the cell in the cell. This happens with an enzyme called hexokinase. It phosphorylates the glucose to the 6th position to give you glucose 6-phosphate. This glucose 6-phosphate has several different potential fates, including the pentose phosphate pathway, glycogenesis, hexosamine pathway, etc. If we want to commit it to glycolysis, we need to do something specific to glycolysis. We can do this by phosphorylating the 1 position to give us fructose 1,6-bisphosphate. However, we need to free up the 1 position in order to do so. So phosphoglucose isomerase is going to shift things around. It's going to free up a hydroxyl group at this end that can then go get phosphorylated by phosphofructokinase to give us fructose 1,6-bisphosphate. Now we have the 6-carbon compound with these phosphate groups on both ends. We can split the compound in half to give us 2 3-carbon compounds, each of which is phosphorylated, trapped in the cell, and interconvertible. Fructose bisphosphate aldolase, typically just referred to as aldolase, is going to do that splitting. It's going to split this 6-carbon molecule into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate can continue straight on. Dihydroxyacetone phosphate can get converted into glyceraldehyde 3-phosphate and continue on. So everything that's happening down here is going to be happening 2 times. Once directly with the glyceraldehyde 3-phosphate that you generated, and once with the glyceraldehyde 3-phosphate that you generated from the dihydroxyacetone phosphate that you converted into glyceraldehyde 3-phosphate. So you can think of doing this path once, and then doing it again with this second molecule. Triose phosphate isomerase does that interconversion between these two pieces. And the glyceraldehyde 3-phosphate, we need this one to carry on because what we're going to do is we're going to oxidatively phosphorylate it. If we think about an aldehyde, we have this carbon, we've got a carbonyl, and it's attached to a hydrogen. We can oxidize it and phosphorylate it by adding an entire phosphate group, this phosphorus with 4 oxygens, such as we can get from an inorganic phosphate group, a free-floating phosphate group. Rather than taking a phosphoryl group from an ATP and sticking it on here, we're actually taking a free-floating phosphate group. This is also an oxidation reaction. We're increasing the number of bonds to oxygen of this carbon. And since this is oxidation, we're going to be using NAD+ and generating NADH. That NADH can later be cashed in for ATP if we're able to do the electron transport chain and oxidative phosphorylation. If we can't, then we're going to have to regenerate this NAD+ that we need, such as by anaerobic respiration using lactate fermentation. But for now, we generate this NADH, and we've generated this 1,3-bisphosphoglycerate. This is going to be a highly quote-unquote energetic bond that we can then use in order to transfer this phosphoryl group onto ADP to give us ATP using phosphoglycerate kinase. We transferred the phosphoryl group, but we left this oxygen, so we're still in this more oxidized state. And we're in the state that we can now convert into a really awkward state in 2 steps. Our goal is to make this phosphoenol pyruvate, where we have this enol, where we have this double bond between 2 carbons, and then this bond to an oxygen. This is what we call an enol, and it's highly energetically unfavorable. This would much rather be in the keto form, where the double bond is between the carbon and the oxygen. But this phosphate group is trapping it this way, so we need to kick off the phosphate group in order to form that, and that'll be really energetically favorable. But in order to make this really energetically unfavorable molecule, this phosphoenol pyruvate that we can then use to kick off the phosphor group and make ATP, we need to first get it in the position to do so. So we've got this 3-phosphoglycerate. What we need to first do is move the phosphate group to the middle position, which we can do with phosphoglycerate mutase. We move it to the center position. Now what we're going to do is we're going to dehydrate it. We're going to remove water. We're going to remove So this is not an oxidation reaction. This is just a dehydration reaction. We're losing hydrogen and oxygen rather than just losing hydrogens. So it's not a dehydrogenation, it's a dehydration. And so this is going to not be redox, but it is going to make it so that we've got that enol. And the enzyme that did this was enolase. Now what we have is this phosphoenol pyruvate. And what we can do now is we can untrap it, remove that phosphate group, stick it onto ADP to give us ATP. So we generate an ATP, and we are left with pyruvate. And the enzyme that does this is pyruvate kinase. We've generated an ATP and an ATP, and we use 2 ATPs up here. So you might be thinking we didn't make any ATP, but don't be fooled, because remember we did this part of the pathway twice, and so we on net generate 2 ATP. We also did this part of the pathway twice, so we did 2 NADH. And if we're using the electron transport chain and oxidative phosphorylation, we can get the electrons from the NADH into the electron transport chain using the malate aspartate shuttle or the glycerol 3-phosphate shuttle. We can't take the NADH directly into the mitochondria, but we can sneak its electrons in. If we go through the malate aspartate shuttle, we get 2.5 ATP per NADH. And if we go through the glycerol 3-phosphate shuttle, we get 1.5. But the glycerol 3-phosphate shuttle is faster. And so sometimes your brain and your liver use that one instead of the malate aspartate shuttle. But that's just a technical detail. At the end of the day, you need some way, though, to regenerate the NAD+, and so anaerobic respiration provides an alternative. If you want to go the other direction, you can't simply reverse these steps, the hexokinase, the phosphofructokinase, and the pyruvate kinase steps. So in gluconeogenesis, you have to take detours. You use pyruvate carboxylase and pep carboxykinase in order to quote-unquote reverse this last step, this pyruvate kinase step. You use fructose 1,6-bisphosphatase 1, FBPase 1, in order to reverse this fructokinase step. And you use glucose 6-phosphatase in order to quote-unquote reverse this first step, the hexokinase step. These steps are going to be inversely regulated mostly in glycolysis and gluconeogenesis. So there you go.